Hi, welcome to the second part in the key concepts of experimental design. In the last piece, I introduced the idea of natural and controlled experiments and mentioned the 5 key elements when it comes to all experiments. Remember, in these pieces, I'm not trying to give you a thorough, exhaustive set of notes on how to conduct every experiment that you can think of. But what I've tried to do here is synthesize the 5 big ideas that you need to have in the front of your mind when you are considering your own experiments, or more relevantly in this course right now, when you are examining someone else's experiment, or maybe a Shinrin-yoku experiment. When you, for example, examine the paper I have sent to you, these 5 elements are elements that definitely sit within the marking rubric. And so you definitely need to be thinking about these things when you conduct your methodological critique of your paper. Remember, the critique is based upon 2 things. A structural critique of the theoretical framework or conceptual framework used throughout the paper. And for content on that, see the academic writing content that's online. This part focuses on methodological constraints. But also bear in mind that this is not the last piece of content focused on methodological issues associated with experiments. In the second half of the course, I will deliver more content focused on experiments, but this time, experiments focused on people. Look for that in the second part of the course. All right, so let's get into these 5 big elements. Number 1, replication. We've heard about replication before and we know what it means. It means to measure more than one thing, to generate a copy, if you like, of something, to produce more than one outcome. So when we replicate in a study, we make multiple observations, like this little baby eucalypt here was joined by many others of the same species in different families, but we replicated within families as well so that we could produce metrics associated with this family, but we could also express error associated with that family in the greenhouse. Bearing in mind that even though we're in a greenhouse setting, variability exists between individuals because of genetics, but also because of the position they find themselves in the greenhouse. We need to replicate and we need to do it always. Without replication, we can't generate key statistics, like a confidence interval, for example. A confidence interval is based upon, as you remember, T. To generate T, we need degrees of freedom, n minus 1. If you have one observation, you can't calculate a T. If you have one observation, you can't calculate a standard deviation. And a mean doesn't mean an awful lot if you've only got one observation. Without being in a position in which we can express error, then our data aren't really all that meaningful. So, if we are interested in particular in ecosystems or systems in general that are highly variable, like ecological or human systems, exactly the thing that we have in mind in this course, well, we definitely need to engage in replication, and indeed in those systems we need to increase our replication so that we have a better handle on the error. So, is it just enough to throw in more observations? Do we just need to make lots of observations about these plants to then say something about different families? Well, we have to be careful about something. And we have been careful about it so far, but perhaps it's just been a little too implicit. This is the moment in which we become explicit about a particular problem when it comes to replication, and that is the idea of replication scaling. Now, when I say the term replication scaling, I'm not talking about how many observations we have I'm talking about a conceptual issue here. The concept of ensuring that the replication we have is appropriate for the question we've asked. That is, for example, if we wanted to compare between aspects on Black Mountain, then we need to make sure that we replicate at the scale of transects, not at the scale of plots. So let's say, for example, we wanted to compare between north and south aspects on Black Mountain. It would not be sufficient for us to go to one ridgeline and put in multiple plots down one north side, multiple plots down one south side, and then use the mean and standard deviation and so on from those plots to compare between them. Why not? Well, because we only have one north side and one south side. And that is the scale at which we're asking the question. So that is the scale at which we need to replicate. If we observe a difference between the mean of the plots on the north side and the mean of the plots on the south side, all we can say is that between these two sites, there is a difference. And that's not really all that informative. If we wanted to compare between aspects, we would need multiple ridgelines across which we place multiple transects so that we have multiple north transects and multiple south transects, but they must be on different ridgelines. You can't put two transects on one ridgeline and then say, well, I've got different transects here and I'm going to treat them as separate sampling or experimental units. That doesn't work. We have to think about replication at the scale of the question. How many of your Shinrin-yoku papers do that, I wonder? So bear in mind, the question here that we need to answer always in experiments is not just how many replicates, but at what level is the experimental unit. The example I just gave, the experimental unit is at the transect, the aspect, specific aspect level. So we need to carefully think through that. It may not be the scale that we conduct our measurements. For example, you conducted your measurements at a plot level, but you now know that replication cannot operate at that level if we're comparing between aspects. So this slide here then just gives you a bit of text that formalizes exactly what I've just said. That replication must be at the scale of the treatment, and if you have multiple observations within a single treatment, then that doesn't test the treatment effect. It just tests, is this group of data different to that group of data? It doesn't necessarily imply that any specific effect is responsible for that difference. It just says these two things are different. And that's not very informative. So bear in mind, there are many circumstances, both within natural ecosystems and within human ecosystems, within individual people, in which multiple observations cannot be made to then make a statement about the effect of, for example, if you were interested in the effect of fires on ecosystems, you couldn't just go, well, I measured in a burnt area and an unburnt area if there was just one fire, and then you make a generic statement about all fires. That doesn't work. That's just between this location and that location. And this is a problem associated with many studies that compare the impacts of human modification of ecosystems, where comparisons are made between a single reference ecosystem and a single modified ecosystem. Yes, there's replication within those, but there's only one comparison being made, and that's between one group of data and the other. It's not a treatment effect. There is a name for when you get it wrong, and it is called pseudo-replication. You think you've replicated, but you haven't. So point-based estimates, as we will see, of crown cover on a transect, that's not replication, that's pseudo-replication. We need to aggregate all the observations and then treat it at a transect level. And there are many other examples of that. So how do you handle pseudo-replication? Because sometimes, goodness gracious, it's hard to handle. The thing to do, first of all, when setting out to conduct an experiment is to consider the scale of the question. Doesn't matter what you're working on, think about the scale of the question and the scale of replication. Make sure those things match. If you can do that, great, proceed. But what if it's just not possible? What do I mean when it's not possible? Well, think about this example behind me, right? You mightn't be into plants, but you know, I am, and you're kind of stuck with me for a little bit. But this is a good example of the problem. So when we conduct experiments in greenhouses, you know, there's a lot of physicality to it. You know, these 7-litre pots, when they're full of water, you know, they're approaching 7 kilograms and you move them around all day. But aside from that, they're big. They take up space. And there isn't an endless amount of space available for us to conduct these experiments. And at times, if we're interested in genetic responses to different treatments, we need a lot of replication within an individual genotype to actually measure its traits appropriately. Well, that's just not possible. We don't have huge greenhouses here, the size of a commercial greenhouse, and we certainly, even if we did, we couldn't inject that full of CO2. It's just too expensive. So we have limited space and limited facilities. If we were to conduct these experiments recognizing the issue of replication seriously, what we would need to do is to not have multiple plants in one room with CO2 and then plants with non-CO2 in the other room. Because if we do that, we incur pseudo-replication. Why? Because we have 2 rooms. This room might be just different to the room next door to it. So we're not necessarily testing a CO2 response, we're just testing the difference between 2 rooms. And that's not that interesting. That doesn't tell anyone anything. But to replicate at the scale that we'd need to, we would need many, many greenhouse rooms, and they're just not available. It's too expensive. So what do we do in those circumstances? Well, here are a number of things. There are solutions to these problems. One, we can repeat the experiment several times. That counts as replication. We do the same thing over and over and over and over. That is experimental life. Repetition is what experiments are about. And if you're not up for that, then you're not up for experiments. The alternative, not as good, but it still works pretty well. We can move the treatment and control. Let's say we have a treatment in here that's elevated CO2, or we've drowned it or whatever, and we have a separate room next door. We can, partway through the experiment, we can switch the plants from room to room. Yes, we need to change the treatment so that one room that was the control room becomes the CO2 room and vice versa. And we don't need to do it just once. We can switch backwards and forwards so that the plants or the subjects are experiencing the same conditions over the course of the experiment. That's quite a lot of work. Bear in mind, in this room, there are many plants that weigh at least 7 kilograms, and all of them would need to be moved very, very quickly at the same time. And we don't want to impact on the experiment itself. So we don't do it in the middle of the day. We come in to move the plants when it's dark, when they're inactive. Move them around, and then when they wake up, they're in a different place. It takes a lot of effort and time. Even within a greenhouse, we need to rotate plants. And the same is true, perhaps for other experimental conditions with animals and also other subjects in a lab. So there are strategies around it, the problem of pseudo-replication, but they need to be taken on very seriously. All right, number 2 in our list of 5 key things when you're designing an experiment is the application of a control. We know that if we're interested in testing a treatment response or we're going to modify something, that we must also measure what a controlled circumstance is. So people who, for example, haven't been out in the forest hanging out in a hammock having a nice time, or areas of land that have been fenced off so we can see areas that have not been attacked by animals and eaten. We need to have that control. Now, you probably already know about that, right? You have a treatment, you have a control, and you compare between them. Remember, as you probably are already aware, that what we're trying to do is eliminate anything that is an experimental artifact. Not just see what a plant or anything else can do in the absence of the treatment. We need to eliminate the artifact of applying the experimental treatment, not just the treatment itself. So what does that mean? So let's think about the example that I gave before about mycorrhizae. The way that we inoculate plants with mycorrhizal associations is we let them germinate, and then we have the spores and we water the plant with a slurry of the spores. Now, in that spore mix is a set of organic nutrients that the plants also benefit from. So that means that a plant with that is given a mycorrhizal association also benefits from a little nutritional kick. That is an experimental artifact. We weren't actually interested in the nutritional effect of applying the mycorrhizal spores, but we got it anyway. So any benefit, any change in the plants after the application of the mycorrhizae might, in part, be a product of that artifact, the nutritional artifact. We have to control for that. How do we control? Well, we take some of those spores and we put them in an oven and we cook the crap out of them, basically. We kill the spores. And then we take those spores, mix up a slurry, and we give that slurry to the plants that are the control. So all plants receive a slurry. One of them is alive and the other is dead. Anything we do that is a treatment, if we are taking individuals out of the setting in which we are going to apply a treatment. So let's say, for example, we're going to apply some treatment to some animals out in the wild. We catch them, we apply the treatment, whatever it is, and then let's set them loose. What we also need to do is capture the control animals, weigh them and do whatever, and then hold on to them or manipulate them pretty much the same way as we would if we were going to apply the treatment, but then just not apply the treatment. You're probably familiar with the idea of controls in the use of placebos. We've heard about the use of placebos in drug trials, for example, where people are given something that takes the place of the actual treatment itself. The idea here, people, plants, ecosystems, whatever, the idea here is that the control individuals are treated identically to the treatment group, with one exception, and that is the treatment itself. Any variation in the way they are treated is an artifact and may affect the outcome. Artifacts are to be avoided. Why? Because they lead to confounding associated action effects. The associated action is, for example, anything that we do that is involved in applying the treatment but is not the treatment itself. Associated actions have effects, and we need to control for those. Associated actions, for example, in ecology, might be if we want to fence out an area that keeps kangaroos out or keeps grazing out entirely. If we get the type of exclusion wrong, if we're just interested in excluding kangaroos, we need to use a mesh that's quite big that will let rabbits in. If we want to exclude all animals, then we have to use fine mesh. That excludes rabbits as well. We need to consider the associated action. An example that I'm familiar with are these things. Believe it or not, this is just near Sydney, where these structures are set up to study the impact of enriching the air with CO2, a free air CO2 enrichment trial. But the action of setting up the structures changes other attributes of the environment, changes the humidity and the light and all sorts of things that then impact upon growth. The action of establishing the trial has an associated effect that is in addition to the treatment. We need to be aware of associated action or artifacts in our trials. You can see I forgot the word there for a second. All right, number 3 in our list. Only 2 more after this. Number 3 in the list is randomization. We have talked about randomization in the context of the central limits theorem. You know that it is important and that in the absence of randomization, we cannot assume that our sample data are generalizable. They're not representative of the population that we're interested in. So that means we don't just randomly select individuals, but we also randomly assign individuals to treatment or control. Now, this can produce a problem if we conduct our study in a completely randomized fashion. And many of you saw that problem, and gee, that blue gum data, it just keeps giving. Many of you saw that problem in the trees that you were allocated in week 1. You were allocated trees randomly. Many of you had duplicates. And that's a natural outcome of random sampling, that you can end up with an aggregation of randomized treatment or control individuals that are concentrated in one part of the population. And if you follow through on that, you'll end up with a very skewed perspective of the population. What that reflects, and that is a product, much like the Dilbert cartoon behind me, that is a product of randomization. And it is referred to as an interspersion problem. So this example here of the random number generator that keeps spitting out the number 9, which is a possibility, that is poor interspersion. All of their random outcomes are aggregated here in the most ridiculous sense on a single value. But it can happen in our own work, where we end up with a cluster of observations over a narrow spatial or temporal or genetic or whatever spread of our population that we're interested in. And that can, even though it's random, produce a problem. How do we handle that? Well, we need to, at times, look at the interspersion of the random allocation of individuals to treatment or control, or the random identification of locations, individuals that form part of a study. We need to, in a subjective fashion almost, make a decision about what level of interspersion we are happy with. Now that's a very subjective process, and naturally you should be wary of that. Any approach that seeks to achieve a specific interspersion approach risks introducing bias of its own. So where do we go there? That is a very difficult line to walk, and only one that can be handled with experience over time. An alternative is to systematize the way in which plants are arranged. And one approach is to use this thing called a Latin square approach. In a very designed spatial array, plants are placed in rows and columns, or individuals in rows and columns, and an individual never appears in the same row-column combination. It's like an experimental Sudoku. It's exactly an experimental Sudoku, but instead of a 3 by 3, we might have a 10 by 10 square. Now we might do something different, something that is nicely referred to as haphazard allocation. That is, we do it ourselves. We try to be random, we spread things out and sort of mess it up and look so it looks random, but it looks random to our eye. Of course, the trick to something that's random to our eye is we tend to see things as random when they're fairly uniformly distributed. That's not random, right? That is a pattern that suits our eyes. That's a haphazard allocation. And it's widely used, but again, it's one that we need to approach carefully. The fourth thing in our list of our 5 big things is independence. Now, what does independence actually mean in this case? Well, we have an independent variable, a variable that explains variation in the dependent variable. We're not talking about that. This can get confusing because independence is something that we talk about in different frames. Independence in this case is that our individual experimental units, our individual people, or our plants, or our observations themselves, do not affect each other. They don't have an impact on what another's experimental unit will express in terms of its quantitative value. Now, how might that be true? Well, if you have people in an experiment and they talk to each other, so a person comes in, you conduct a survey or an interview with them, and then they walk out and they go, hi, how you going? I didn't know you were part of this. Yeah, you know what they do in there? They do this. No. That's not independence. Individuals need the space to respond to trials, surveys, experiments independently of others. Anything that risks interaction between experimental units is a problem. So we can think of it in other attributes. Here's a favourite data set of mine, or a type of data set. This is a tree ring data set. But this is not specific to tree ring data. This is a temporal sequence, a time series of data. These are ring width data, but we could be looking at any time series. A person's state of mind over time, anything at all, ecosystem attributes, animal, anything over time. Note in particular that for a tree at least, look at the start, there's always this decline from fast growth down to slow. So the rate of growth here depends upon exactly where in the sequence you are. And in particular, it depends upon what the value was in the preceding year. Time series data are so very, very, very rarely truly independent. Time 1 to time 2 almost always carries some persistence into the next measurement. So time series data, for example, if we run an experiment that involves measurement of an effect over time series data need to be handled very, very carefully. Now, we don't cover time series data in this course. There's just no way we could get to that. But be aware that time series data come with that problem, and it is independence. It is, of course, evident in other data. Let's say we use a piece of equipment, maybe a really fancy piece of equipment like a mass spectrometer that has gas lines running through it. Now, when we use a mass spectrometer, we're analysing the composition of the gases that pass the magnet. Now, in the gas stream itself, those gas streams can develop a memory. They don't remember what the value was before, but they retain some of the chemical signature of the samples that have come before. Meaning, again here, a time series, but it's not formally expressed as a time series, but when we measure things in sequence using a piece of equipment, we need to be careful about what impact previous measurements might have on our ability to measure the next thing. So Watch for measurements that have a memory effect. Yeah, just some points around time series data. The next in our list of the things to be aware of in experiments is blinding. Now, I have referred to blinding in a way earlier. Remember, I referred to placebos, and we're aware of what placebos are, and that is when a false treatment is applied in the place of a genuine treatment. And we do that to ensure that those associated action effects are eliminated from the experiment. Now, the process of applying a placebo does not work if the individual under study knows it's a placebo. The subjects need to be blinded to that. But that's only one level of blinding. In research, we try to blind at many levels. We try to label our units, our experimental units, in a way in which we don't know what species they are, what family they're from, whether they are treatment or control themselves. We want to, as experimenters, conduct our experiments as blind to what the individual is as possible, because we are also a source of bias. Maybe we behave ever so slightly differently with a treatment. We talk to them in a more kind way, or we are less rushed when we're doing the measurements with them, or we have a tendency to round up when we're, looking at a treatment. We need to be careful about blinding of ourselves as well. Indeed, many researchers now, or research teams, separate the experimental team from the data analysis team. Now, that's simply not possible in most cases. Most research teams are too small for that sort of thing, but in global research teams, that's the type of thing that we do. So, remember, placebos work, but the experimental unit needs to be blind to the treatment and the placebo, and ideally the experimenters too. Especially if it's a survey where you're asking them questions, the experimenters must also be blinded as well. True blinding happens both sides. Double-blinded studies. Something to be aware of, and we will come back to this issue, and it is really a significant problem when we deal with people in surveys, for example, is the issue of demand characteristics. Demand characteristics are when participants are aware, perhaps, of what the hypothesis is, or they're expecting something to happen, and then they behave in a way that, well, artificially generates the effect, or the absence of the effect, on their own. Now, this is something that is particularly true if people become aware that they've been given the treatment. Of course, it is what placebos play upon, the idea that we give you something and then you believe that you've had the treatment, and then that might generate some kind of response. Demand characteristics are the characteristics that when people are, or subjects are aware of what has happened or is about to happen, when those characteristics lead to a systematic change in the person, the individual's behaviour. And it's not just people. Recent studies out of Adelaide have indicated that plants too have the capacity to learn and anticipate treatments, presenting a problem for anyone like me who does mean things to plants. Well, those are our 5 things. But before we move on, when it comes to blinding, especially for people, There's one strategy that is particularly widely used, and when it comes to animals and plants, of course, it's relatively easy to consider the ethics behind it. But when it comes to people, the use of this strategy is very, very controversial. People, of course, are always trying to work out what a researcher is actually interested in. What are they testing? And they can't help themselves. They're always trying to guess what an experiment is about. Even me, occasionally I participate in psychology student experiments, and I can't help myself while I'm being a subject. I can't help but wonder what the experiment is about. How do experimenters stop you from doing that? How do they trick you? How do they keep demand characteristics and independence central to their studies? Well, this thing called deception, where researchers use, in effect, a lie to tell the subject what the experiment is about. They make them believe that the experiment is about something that it is not. And then, under that pretense, they collect data that is specific to their experiment of interest. One of the most famous examples of deception, and in particular its misuse, is the Stanley Milgram experiment. Now, I have not included any links to that in this lecture in particular. I will put a link online subsequently, but be aware that the Stanley Milgram experiment was particularly traumatic for the people who were involved, and it has become an example of exactly what we should not do when we run experiments with people. So how can deception work? Because sometimes it might be necessary, and you might see this is the case in your Shinrin-yoku papers. How can deception work in an ethical, appropriate way? What comes to mind are two examples. The first example is a study that I'm aware of that was conducted in Harvard of subjects' tendency to consume energy-rich, unhealthy foods when they are placed under stress or given short time periods in which to respond. Now, the researchers brought their subjects in, conducted what they pretended was a stress test, and during that stress test got the individuals to select food items that looked delicious from a screen. Yeah, I'm under pressure. I want a cookie. I'm feeling quite relaxed. I wouldn't mind a pear. That kind of thing. That was all deception. And at the end of the experiment, depending on how difficult the experiment was, the experimenters got the person up and said, okay, well, we're just going to take you down the hallway now, where we just need to finalise some paperwork, and then we'll send you on your way. They got up, walked out the door, and at that moment, a grad student walked past them with a plate of food, and they just said, and this was the person who greeted them at the start when they came in, just said, hi, hey, thanks for helping us out. We've just got some food. Here, do you want something? And without thinking, the subject would reach and grab for a pear or a cookie, whatever it was on the plate. That was the experiment. The moment afterwards. Now, the experiment was about the same thing as they had said it would be, but it was the moment in which the data were collected, not the treatment itself, that varied. So deception doesn't have to be an outright lie. The experiment is about this and then we do something else. But you do not need to disclose what moment the data will be collected. That is an ethical approach to deception, deceiving the subject about the measurement moment, not the measurement itself. Anyway, that's the end of our content on experimental design. Remember, this content is particularly relevant to your Shinrin-yoku studies, and there will be more content focused on experiments and surveys of people in the second half of the course.